The next optical instrument we're going to look at is the eyeball. The human eye, the insect eye, most eyes work basically the same. For novel focus, the eye focuses light and produces an image back here on the retina. The essential parts of the eye are the cornea, and the light passes through this transparent structure, and this is where most of the refraction happens because this is where we're going from air to liquid. So most of the bending of light actually happens at the cornea. That liquid is called the aqueous humor. It's a clear liquid behind the cornea. Sometimes it has some floaties in it, but generally it's clear. The pupil is actually the opening in the eye that the light passes through, and it's a variable aperture, very much like a camera has a variable aperture. And then a crystalline lens right here behind the cornea, behind the pupil. So the most refraction takes place at the outer surface, and the cornea is covered then also with a film of tears to protect it. The iris is the colored portion of the eye around the pupil. It is basically muscle. It's a muscular diaphragm that controls that pupil size, and it regulates the amount of light entering the eyeball. So if you have very dim light, you can have a wide open pupil. So this is for dim, and for very bright light, you can have a very closed down pupil. So the F number for the eye ranges from about 2.8 to 16 generally. And what happens is this lens cornea system is able to produce a real image on the back of the retina, as it is a converging lens system. So the back surface is called the retina, and the retina is covered with a whole bunch of cells. These cells are called rods and cones, and then the optic nerve comes out of the back of this. You can find online many ways to be able to figure out where your blind spot is, and the blind spot is the spot where your optic nerve comes into the back of your eye. Your physiology does an amazing job on covering that up. Then it's not until you actually do an experiment, it's like, whoa, yeah, that thing just disappeared. It's a dark spot, a blind spot. So we have rods and cones, and the amount, so our eyes have a resolution that we can't see below. We'll talk more about resolution at the end of the chapter, but if this sensor is activated and this one is off, it's basically the minimum resolution. We have pixels in the back of our eyes because there are cells, individual cells, picking up these wavelengths using a chemical process and then converting that into an electrical signal. And so if you get into it, the process is quite fascinating. There's lots of proteins involved, and you can kind of get a haloing effect, or if you look at one color for too long and then look at the complementing color, those cells have been overstimulated and you get some interesting effects. There's lots of optical illusions that you can do. So how does the color vision work? We have three main cones. The rods, while they don't see color, have the peak sensitivity at 498. It's a kind of a bluish green, but our physiology doesn't translate that into color. It just translates that into bright or dark. And so we have three basic cones. You have your red, your green, and your blue cone. So it's no wonder why that when we try to display color on an RGB display, that we in fact put out those wavelengths to directly stimulate our physiology and make all the colors, because all the colors can be made of these three, because those are really the only three colors that we sense fully. All the other colors are made up of combinations of these three colors. And so the red cone, or the S, M, and L, have a different range of sensitivities. And so these two overlap, and the most common one are these long ones in the back of your eye. So the color, here's an SEM of some of the rods and cones. So these are the color cones, and the rods are the green ones, so rods, and the blue ones are cones, and they're covering the back of our eye. You can look at the experiments that they can do to tell which cones are stimulated by which color, but basically involves staring at a a monochromatic picture for a long, long time, and then looking at the back of your eye and seeing which cells are stimulated.